Click the bell icon and subscribe to our channel so that you will not miss any classes.
Hello students, welcome to the class. The third phase in the mitotic cell division is anaphase. We studied from the last class that at the metaphase, I am writing here right now the cell which is present in the metaphase, where there is a metaphase plate formation. All of the chromosomes are arranged at the equatorial and they are being attached with the spindle fibers. So this is the metaphasic chromosome. Once the cell enters into anaphase, it is marked by the splitting of centromere into two. That means each chromosome is consisting of a centromere, and each of the centromere they split into two like this. So suppose it is a cell in anaphase. In the early stages of anaphase, what happens is, here is the centromere and this is these two are the sister chromatids, and from here one spindle fiber attached and from here another spindle fiber attached. When the cell enter into anaphase, in the early stages what happens is, the centromere will split into two like this. This is one split part and this is another split part. So from here one spindle fiber and from here another spindle fiber, and these are the chromatids. So here the centromere has split. So this is marked by the anaphase. So the beginning of anaphase marked by the splitting of centromere of all the chromosomes, and that too at a time. At the same time, all of the chromosomal centromeres will split into two, and they are found to be the centromeres of future. That means the cell will split into two cells, right? In that cell, these two are the separate centromeres. Even though they are arising from the same centromere, they develop into individual chromosomal centromeres. That you need to keep in mind. So now this is the beginning of the anaphase. So we shall represent it now right here. So since we have taken 4 chromosomes, so here is the first chromosome which has split its centromere, and here one more chromosome which has split its centromere, and one more has split its centromere, and another. So I'll write here. So the 4 chromosomes have split at a time, and after splitting of the centromere into two, the centromeres will start moving towards the respective poles. That means this centromere will start moving in this path, and this centromere has started moving to its centriole. So this is because the contraction action of the spindle fiber. Actually, what is the exact reason for this movement is not clear till today, but there are various theories. They suggest that the contraction action of spindle fibers or the microtubules is one of the reasons for the opposite movement or pole-wise movement of the centromeres. So that is the one theory it suggests. Once the contraction of these spindle fibers happens, which will result in the various changes which is taking place in the cytoplasm. That is, the refractability of the cytoplasm will changes and the viscosity will changes, and there are various movemental changes which takes place in the cell cytoplasm. That is also may be the reason for this movement. That is the suggested theories, but actual reason is unknown till today. So this is the beginning of the anaphase, and as the process continues, here the pathwise movement and here is the pathwise movement which is taking place. When it continues, here the chromosomes have started moving towards the poles. They started moving towards the opposite poles like this. And remember, these are the chromosomes of the future daughter cells. In future, there will be formation of two daughter cells, right? And in these two daughter cells, these are the chromosomes. This is the 4 set of chromosome, and this is the 4 number of chromosome in one cell, and these are the 4 number of chromosomes in another cell. So these are the, even though they are split from a single chromosome, but they will be the chromosomes of the future daughter cells. Now, once the movement has started, the movement is very peculiar. The movement of the only centromere will take place. That means we know that the spindle fibers will be attached to the kinetochore at one place, right? So the traveling of only the centromere will take place towards the pole, but not the arms of the chromosome or the chromatids. The chromatid arms will not move. What will happen? They will be dragged along with the centromere. That means only the centromeres will be moving towards the opposite pole, and the arms of the chromosomes will be radiating in the cytoplasm. In the cytoplasm they will be floating. So that's why we will be writing the opposite or the opposite direction. We are writing the chromatid arms in the opposite direction because only the centromeres are moving towards the poles. The arms are radiating in the cytoplasm, and depending upon during this moment, depending on the position of centromere, depending on the position of centromere in chromosome, during this anaphasic movement. This movement is called anaphasic movement. During the anaphasic movement, depending upon the position of centromere in a chromosome, the chromosomes will appear in different shapes like V shape, J shape, L shape, I shape. This is on what basis? On the basis of the position of centromere in the chromosome. That the different shapes are visible only during the anaphasic movement, not all the time. Anaphasic movement means movement. Anaphasic movement means because of the contraction of spindle fibers, the centromere will move towards the poles with the radiating chromatids. So during this anaphasic movement, depending upon the position of centromere in a chromosome, the chromosomes will appear in different shapes. Suppose if the chromosome has its centromere at the center like this. At the center, then this type of chromosome is called metacentric. Meta means center. This type of chromosome is called metacentric chromosome. During the movement of metacentric chromosome, during anaphasic movement, the chromosomes will appear in V shape like this, or you can write it ulta. So they appear in V shape, metacentric. And one more, if the centromere is present slightly away from the center, not exactly at the center, slightly away from the center, then it is called submetacentric. Then this is called submetacentric. And here we could observe the shape of the chromosome L shape. It will appear in L shape, alphabet L. And if the chromosome centromere is present slightly away from the end, here I am referring slightly away from the center, here I am referring slightly away from the end, then this is called acrocentric chromosome. And during the movement of acrocentric chromosome, the chromosomes will appear in J shape. They appear in J shape. And the fourth type, if the centromere is present at the end of chromatin, like this, then it is called telocentric. During the movement of telocentric chromosome in the anaphase, it appears in I shape. So these are the different shapes which are seen during the anaphasic movement. And by the end of anaphase, there is a formation of two groups of chromosomes at each pole. At one pole, one group of chromosome, and at another pole, there is another group of chromosome. And remember, these two groups of chromosomes are the chromosomes of future daughter cells. And by the end of anaphase, there is a disappearance of chromatic arms. That means the chromatic arms. That means the chromatids will start disappearing, but the operators, that is the mitotic operators, will remain as such. Mitotic operators will be formed at the end of prophase. That mitotic operators will sustain. The mitotic operators will sustain for some time. Which means, I am referring here some time, which means after that it will also get disintegrated. All the things will get disintegrated, but by the end of anaphase, there will be a disintegration of chromosomes, but the mitotic operators will sustain for some time. After that, this will also get disintegrated. So these are the events during the anaphase. The key events you should remember is the splitting of centromere at the beginning of anaphase and the poleward movement of centromere with the radiating arms. During this moment, we could observe different shapes of chromosome depending upon the position of centromere in a chromosome. So there are four possible positions of centromere in a chromosome. Depending on that, the chromosomes will appear in V, L, I and J shapes. That too during the anaphasic movement only, not during any other times. So that's all about the anaphase and the events during the anaphase. We shall continue with the last phase of mitosis, that is the telophase, in the next class.